In this video, I am discuss about the isotonic solution, hypertonic solution, hypotonic solution. What is the tonic? Tonic means tension or concentration. So one by one detail discuss about that. First, we start about the hypertonic solution. Okay, what is hypertonic solution? Hypertonic solutions means the concentration is very more. Okay, so we take a beaker and in this beaker we add solution. The solution is the 2% NaCl. 2 gram of NaCl in 100 ml of water. So this is the 2% NaCl. Okay. So the concentration of solute is very more and the concentration of water is very less. Okay. So this is the water and this is the solute, and then this is the hypertonic solution, and in hypertonic solutions we put a red blood cell. Which cell? Red blood cell. And the outer layer of the red blood cell called the semi permeable membrane. Semi permeable membrane means which allow only water, not allow solute. So this is semi permeable membrane. Okay, so the concentration of solute inside the cytosol is very less, and the water is very high. Okay, so during osmosis the movement of the water from high concentrations to low concentration or low solute concentration to high solute concentration. Yes, so the movement of this water from the cytosols to the solution, and this is the osmosis. So in this condition the cell become a shrink, and when the cell become a shrink, the cell is died. This process called as crenation. So this is the hypertonic solution. And the next is the hypotonic solution. In the hypotonic solution we add water. Okay, so the concentration is very less, means the concentration of solute is very less and the concentration of water is very high. That's why it is called as hypotonic solution. And when we put the red blood cell in hypotonic solution, this is the water and this is solute. Okay, inside the cell means cytosol, the concentration of solute is very high and the concentration of water is low. So the movement of the water from the high concentrations to the low concentration, or from lower solute concentrations to higher solute concentration. Okay, so the movement of the water from the solutions to the cytosol, and then the cell is enter inside the cell, so then become a bulges, and when the cell become a bulges then suddenly it burst. So this process called as hemolysis. Lysis means breakdown and hemo means blood, so the breakdown of the blood. Next is the isotonic solution. Isotonic solutions we add 0.9% NaCl, means the 0.9 gram of NaCl in 100 ml of water. Okay, so this is the solute and this is the water, and in this solution we put red blood cell. Okay, water, solute. So then the solute in cytosol and the solution both have same. The concentration of solute in cytosol and the solution both have same. So the water movement from the solvent to solution or solution to solvent. So in this condition there is a no change in shape and volume of the cell. Why? Because both have same concentration. That's why there is no change in shape and volume of the cell. Okay, so this is the isotonic solution. What are the conclusions about these three solutions? The conclusions behind that, when the cell we put in the hypertonic solution, the concentration is very more, so the cell become a shrink and then a die. This is called as coronation. And when we put the cell in the hypertonic solution, means the concentration is very less, so the cell become a bulges and then burst, so this is called as hemolysis process and cell die. And this is the isotonic solutions where the cell live. Why? Because the cytosol and solution both have same concentration, that's why there is no change in shape and volume of the cell. So inside the human body is made up from the cell, and the cell is around the fluid is present called as interstitial fluid. So interstitial fluid and cytosol both have the same concentration, that's why they are live. This is called as isotonic solution place. So this is about the solutions.